I'd like to thank the organizers for this chance to present. So my group has used the CBTN data in several publications over the last couple of years, but here I'm going to talk about our work in somatic structural variants, or SVs, in pediatric brain tumors. As background, cancer genomes are characterized by widespread somatic genomic rearrangements. Each SV involves two breakpoints representing different genomic coordinates from the unaltered genome being joined together in the cancer genome. Classes of SVs may include deletions, insertions, inversions, tandem duplications, translocations, and more complex rearrangements. Pictured here are the different types of genomic rearrangements that may occur in cancer. And each of these types of rearrangements may contribute to this phenomenon of SV-mediated cis-regulatory alterations. And so for each gene, there is the non-coding region surrounding the gene. That would be the regulatory region that influences how that gene is expressed. But in the event of somatic genomic rearrangement, this regulatory region may be altered in such a way that it alters the expression of the nearby gene. And so somatic SV breakpoints outside of genes may result in altered cis-regulation. This is in contrast to the scenario at the bottom, whereby somatic SV breakpoints within genes may involve gene disruption or gene fusions. And there's been a lot of attention on gene fusions in cancer, but we are also here interested in this phenomenon of altered cis-regulation. And there are a number of known mechanisms associated with SV-deregulated genes. More mechanisms likely remain to be discovered. We have at the top disruption of topological associated domains, or TAD disruption. This has to do with how the DNA folds, and rearrangement can break this folding in places and open up the DNA to transcription that normally wouldn't be open. Enhancer hijacking in the lower left. An enhancer from a different part of the genome may be moved next to a different gene via rearrangement and then increase the expression of that gene. And then a few years ago, my group reported this phenomenon whereby the DNA methylation landscape near genes may be altered by rearrangement, and that would alter the expression of the nearby genes. Now, we have studied this phenomenon previously in adult cancers, but these CBTN data offer the opportunity for us to carry out this study of pediatric brain tumors, which we published last year in Nature Communications. And the CBTN needs no introduction to this group, but we were particularly interested in this combined whole genome sequencing and RNA sequencing of 854 pediatric brain tumors. And so that allows us to look for SV altered gene expression patterns. Represented here are the numbers of somatic SVs detected for each tumor sample by histologic type. So you see that tumors of any histology can be impacted by SVs and impacted also by SV-altered expression. By our analytical approach, we could identify hundreds of genes with altered expression associated with nearby SV breakpoints in pediatric brain tumors. Shown here are the numbers of statistically significant genes with an association between a nearby SV breakpoint and altered expression. Here we're looking at 4 different genomic region windows in relation to genes. We can look for SV breakpoints occurring directly downstream of the gene, or within the gene itself, or directly upstream of the gene, or even expanding our search to 1 megabase surrounding the gene. Even an SV breakpoint within a megabase of a gene can influence the expression of that gene via the long-range impact of enhancers, for example. Genes above the 0 axis are those that have a positive correlation between SV breakpoints versus expression, so genes that are highly expressed in relation to a nearby SV breakpoint. Genes below the 0 axis are those that have a negative correlation between SVs and expression. And then here we're also looking at 2 different statistical models, and for our downstream analyses, we use this model that incorporates a copy number alteration, or CNA correction. And these would involve associations that could not be explained by copy number alteration alone. Shown here are the 627 genes significant for any region window examined. The genes in red are those that have a significant positive correlation between SV breakpoints and expression. genes in blue, those with negative correlation. And among the positively correlated genes would include many known oncogenes as well as gene fusion events. And then those negatively correlated would include some well-known tumor suppressors such as NF1, SMARCB1, and CDKN2A. And these box plots would visualize the SV-associated altered expression patterns for selected genes. So here we're looking at TERT, MIB, and NF1. And we have the tumors in each of these plots broken down by alteration class. And the first group is the SV altered group. So for TERT and MIB, which are oncogenes, there are SV breakpoints occurring 100 kb upstream of these genes. And for those tumors, the expression of the respective genes is much higher as compared to the rest of the tumors. And then on the right, the NF1 gene is a tumor suppressor, and there are 7 tumors that had an SV breakpoint within the gene, and that SV basically breaks the NF1 gene, and those tumors have very low expression as compared to the rest of the tumors. We could identify different SV expression associations between pediatric and adult brain tumors. So on this plot, we have on the y-axis the significance of association between expression and SV's breakpoints for the pediatric brain tumor cohort. And on the x-axis, we have the corresponding significance of association in adult brain cancers, looking at the TCGA data sets. And we can see from this plot that there's actually very few genes that were significant in both cohorts. Those would be the genes in the upper right quadrant of this plot, but for the most part, the genes found significant in the pediatric brain tumor cohort were not found in the adult cohort. And so pediatric brain cancers are different from adult brain cancers. They have different etiology and different set of somatic alterations and impacting a different set of genes. We could identify gene fusions in pediatric brain tumors by an integrative analysis method. So just by RNA-seq analysis, candidate fusion events can be identified just looking at chimeric RNA-seq reads. But if we cross that with the whole genome results, we find that only a fraction of these candidate fusion events by RNA-seq also would have an SV breakpoint within one or both genes, and as well as associated with overexpression of one or both genes. And on the right of those, there were 415 gene body SV breakpoint events that we identified that were associated with overexpression of those genes. And the majority of these, when we looked at the RNA-seq chimeric results, could be associated with a gene fusion in pink. I've shown here are the top gene fusions in pediatric brain tumors involving 2 or more tumors, and then many other fusions just detected in one tumor. But no surprises here. You can see the well-known BRAF fusion and low-grade glioma and the RLA fusion and epidemoma. And then there's this long tail of fusions that occur in 2 to 4 tumors. We could identify SVs associated with TAD disruption and enhancer hijacking. So on the left of the SVs associated with gene overexpression, these were highly enriched for SVs that were TAD disrupting versus TAD preserving. So TAD disrupting would cross two TAD boundaries and break the folding of the DNA. On the right would be, we see that SVs associated with gene overexpression also tend to be enriched for what we putative enhancer hijacking events where an enhancer was brought closer to the gene as a result of the SV. These circus plots would visualize SVs involving key oncogenic or tumor suppressor pathways. So the circus plots would visualize the DNA being rearranged and moving from one part of the genome to the other in relation to some of these important oncogenes or tumor suppressor genes. So there would be a breakpoint nearby these genes. And these genes would fall into some general pathway categories, including receptor tyrosine kinase, P53RB, TERT, SMARCB1, MYC, and Hippo. And the oncoprint here would catalog the somatic alterations for genes falling within any one of these major pathways listed. So if a gene within this pathway had a somatic alteration, we would catalog it as such in this plot. And of the 854 tumors we examined, 558 had at least one somatic alteration involving one of these pathways. And when we do the cataloging, we first look for a small mutation in green, and then we look for copy number alterations and then gene fusions. And the last thing we look at in this plot is SV altered expression, and that's the yellow portion of this plot. And the yellow portion here would considerably extend the numbers of pediatric brain tumors somatically altered for critical pathways. And this is an important point to keep in mind. If we just do a conventional analysis, just looking within the gene itself, let's say by whole exome analysis, we would miss these additional cases where there's an alteration outside of the gene that's altering the expression of the gene, and that can only be picked up by whole genome sequencing in the integrative analysis. Now, on a different track, we also were interested in looking at SV burden across these pediatric brain tumors. So higher SV burden would be a greater number of somatic SVs detected within that tumor. And we find here, for example, that higher SV burden is associated with more aggressive pediatric brain tumors. So on the left, we find that progressive or recurrent tumors from a patient on average had higher numbers of somatic SVs detected as compared to the initial tumor from the same patient. So this is a paired analysis on the left. And on the right, we find that those patients for which their tumors had a high number of SVs detected tended to have a much worse overall survival as compared to patients with a lower number of SVs. And both of these analyses would correct for histology. So histology would not be able to explain the associations we see here. Now, when we made a search for somatic DNA alterations that could be associated with SV burden, we came across these Histone H3.3 somatic alterations in pediatric brain tumors, which would include H3F3C mutations. So on these two lollipop plots would represent H3F3A on the left, which is known in, say, high-grade glioma and DIPG. But then on the right, we have these 9 H3F3C mutated tumors, which involve these very distinct amino acid changes, these hotspot changes. And the oncoprint on the left here would illustrate these the mutual exclusivity of H3F3C mutations as compared to P53 mutations or H3F3A mutations. And on the right, we find that those 9 H3F3C mutated tumors have very high numbers of SVs detected as compared to tumors in these other alteration classes, including tumors with P53 mutations. So these mutations are associated with very increased SV as well as mutation burden. And H3F3A has been known in cancer, but I think H3F3C has not really been reported in cancer, or maybe would deserve more attention in light of these findings. So in conclusion, we find that pan-histology genomic analyses reveal SVs to play a major role in shaping the transcriptome of pediatric brain tumors. Recurrently altered expression of hundreds of genes and associated with the presence of nearby SV breakpoints were detected by our methods. SV-mediated cis-regulatory alterations considerably extend the numbers of patients with tumors somatically altered for critical pathways. And finally, high overall SV burden associates with H3F3C mutations. Brings us to the end. I thank you for your time. I wanted to thank the patients, their families, and the CBTN investigators for making these data available to the research community.